Today we are going to read about the antibody structure and the functions of the antibody. So after starting this lecture, we are entering into the immunology, immune responses, one of the another important topics that you should know in the immunology chapter. So in the immunology responses, today we are going to read about the structure of the antibody and the functions of the antibody. So first of all, let us read about the structure of the antibody. Here you can see the small structure of the antibody. So it's like Y-shaped. First of all, I will go in the general explanation of the antibody structure. So it's like, you can see it's like Y-shaped. So it's like this. So you see the total structure connected here. It is the heavy chain and the side are the light chain. So these two are the light chain and the whole structure is the heavy chain. Again, I'm going to explain you. So the structure of the antibody is like this. So once I draw like this, so this is the heavy chain. So this whole portion, of the antibody is heavy chain. So whereas after I draw these two lines, so these two lines are the light chains. So this is the basic concept you should know. So again, I am going to elaborate the structure of the antibody. So this is the light chain and this whole region is the heavy chain. So in the light chain, you can see this is the light chain. So this whole structure is the heavy chain. So FAV region and the hypervariable region, the FAV region is situated in the heavy chain, whereas the hypervariable region is situated in the light chain, whereas the antigen binding site is contributed by both light chain and heavy chain. So again, before going to the elaborate, more explanatory antibody structure, I want you to know some brief nomenclature. That means C is known as constant, the H is known as heavy, the V is known as variable, the L is known as light, and SS denotes the disulfide bond, the FC denotes the fragment crystallizable, the FAV denotes the fragment antigen binding. So this you should know. You should know the C means constant, H means heavy, V means variable, L means light, SS means disulfide bond, the FC is fragment crystallizable, the FAB means fragment antigen binding, the VJ, whenever I say the VJ means light chains hypervariable region, and VDJ. The VDJ means the variable diversity joining gene segments reagent from the immunoglobulin gene. So VDJ means like that. So whenever I say the VH region in the heavy chains means variable region in the heavy chain, whereas the CH1 means the constant region of heavy chain 1. So it is the heavy chain which is constant in it is the first region. The CH2 means the constant heavy chain 2, the constant heavy chain 3. So these are the region. This is the first region, second is your third region, and these first, second, third region are the constant. So this is constant heavy chain 2, constant heavy chain 3. So now let us go towards the more explanatory structure of the antigen. So as I have already said, so this is the light chain. So this light chain is connected to heavy chain by the SS. SS means disulfide bond. So this whole region is the heavy chain. So in the light chain, it has the constant light chain and in the light chain it has got the hypervariable region. So VL and CL means the CL means the constant portion of the light chain, whereas the VL means the hypervariable region of the light chain. It has got two regions. One is VL region, that is the hypervariable region of the light chain. Another is CL, that means the constant area of the light chain. And let us talk about the heavy chain. In the heavy chain, there is the VH region. The VH region means variable region of the heavy chain, and CH regions. There are three regions: CH1, CH2, and CH3. The CH1 means constant region of the heavy chain 1, CH2 means constant region of the heavy chain 2, and CH3 means constant region of heavy chain 3. And here it is the FC region. So this portion only, it is the FC region, that is fragment crystallizable region of the heavy chain. So this region, this whole region, so this whole region is the FAV region, fragment antibody region. So the epitope binds in the heavy chain and light chain area, that binds in the this to heavy chain and light chain area, and the FAB that contains the variable or hypervariable region consisting of light chain and heavy chain recognize the antigen, and the FC region of IgM and IgG fixes complement, whereas the heavy chain contributes to FC and FAB region, and light chain contributes to only FAV region. So you have to understand that. Now let us talk about the FAB region. The FAB region is the fragment antigen binding region, and the FAB region determines the idiotype, the unique antigen binding pocket. That means only one antigen specificity is expressed per B cells. And about the FC region, the FC region is constant carboxyl terminal. It binds the complement. It has the carbohydrate side chains, and it confers the isotypes. So this is the property of the FC region. Now let us talk about the generation of the antibody diversity and generation of the antibody specificity. So generation of the antibody diversity, which is antigen independent. The random recombination of the light chain and heavy chain genes creates the antibody diversity. And the random addition of the nucleotides to DNA during recombination by the terminal deoxynucleotide transferase generate the diversity of the antibody. The random combination of the heavy chain with the light chain also creates the antibody diversity. Now let us talk about the generation of the antibody specificity. So generation of the antibody specificity, how occurs? It occurs by the somatic hypermutation and affinity maturation, and also it occurs through the isotype switching. So these are the two methods how the generation of the antibody specificity occurs. So now let us discuss the functions of the antibody. So function of the antibody is prevents the bacterial adherence. So antibody prevents the bacterial adherence by the neutralization. So it promotes the phagocytosis. Antibody promotes the phagocytosis by the opsonization. And the antibody activates the complement system. It enhances opsonization and the lysis by forming the membrane attack complex on the bacterial surface. So these are the very three important functions of the antibody. So we have described the brief antibody briefly about the structure of the antibody, and we discussed about the generation of the antibody diversity and generation of the antibody specificity. And also we discuss about the various function of the antibody. I hope you understood this topic of the antibody structure and functions. Thank you.